Welcome, friend. Tonight, we're going to spend some time together here on Bedtime Biographies. If you're listening to this, chances are you're already settled in. Maybe the lights are low. Maybe you're lying down, feeling the weight of the day beginning to lift. And that's exactly where we want you to be. This channel exists for moments like this one. Quiet moments, restful moments, when you want something gentle to carry your thoughts forward, slowly, without any effort at all. Tonight, we'll follow the life of Barbara McClintock, a woman who spent most of her life in the quiet company of corn plants, studying things so small they couldn't be seen with the naked eye. In a time when almost no one believed what she was finding was even possible, she was a scientist, a geneticist, one of the most original thinkers of the 20th century. And yet, for a long time, she worked largely alone, in a small laboratory, tending her fields season after season, patient in a way that most people simply aren't. Her story is not a loud one. It doesn't move quickly. It's the kind of story that unfolds the way she herself seemed to live. Steadily, quietly, with deep attention to the small and the careful. So let's begin. Barbara McClintock was born on June 16, 1902. The world she arrived in was a very different place from the one we know today. The 20th century had barely begun. There were no commercial airplanes yet. Most people traveled by horse-drawn carriage or early train. Electricity was still new to many homes. It was a time when the pace of daily life was slower. The sounds of a neighborhood were softer. Children played outside for long hours. Evenings were quiet. Barbara was born in Hartford, Connecticut, to Thomas Henry McClintock and Sarah Handy McClintock. She was the third of 4 children. Her family was ordinary in many ways. A working household, a father who was a physician, a mother who kept the home and managed the children. But Barbara, even as a very small child, was not quite ordinary. She was described by those who knew her as unusually independent. Not in a difficult way, but in a self-contained way. She seemed comfortable on her own. She didn't need constant company or noise or activity. She could sit quietly with her thoughts for a long time. Her mother, Sarah, recognized early on that Barbara was different from other children in this way. And Sarah's response was, for the time, quite remarkable. Rather than trying to shape Barbara into something more conventional, she largely allowed her daughter to be herself. That kind of freedom was rare, and it mattered deeply. When Barbara was very young, just a few years old, the family moved. Her father was building his medical practice, and the family relocated several times in those early years, eventually settling in Brooklyn, New York, when Barbara was around 6. Brooklyn, at the turn of the 20th century, was a lively, busy place. It was growing rapidly. Neighborhoods were dense with families from many different backgrounds. There was noise and movement and the energy of a city finding its shape. But within that environment, Barbara found her own quiet corners. She was a child who liked to think. She liked to observe things. She noticed details that other children walked past without a second glance. She would spend time alone in a focused, absorbed way that felt natural to her, even if it sometimes puzzled the adults around her. She also had a physical side. She was athletic. She liked outdoor games and physical activity, and she was good at them. There was nothing fragile or withdrawn about her. She was simply someone who moved between the outer world and her own inner one with ease. Her father, Thomas was a thoughtful man. He worked long hours as a doctor, but he was gentle and attentive when he was home. He shared something with Barbara that her mother didn't always understand: a tolerance for solitude, a comfort with independence, a belief that thinking for yourself was worth more than conforming to what others expected. It's likely that Thomas saw something of himself in his youngest daughter, and while he didn't push her in any particular direction, his quiet support was something she would carry with her for years. School, when it came, was interesting to Barbara, but also slightly frustrating. She was clearly intelligent, quickly able to absorb information, quick to see patterns and connections. But she found the pace of school often too slow and the rules too rigid. She wasn't a rebellious child. She didn't cause trouble, but she was always slightly out of step with the expectations that surrounded her, not because she rejected them exactly, but because her mind was simply moving in its own direction. She liked to learn. That part she loved. But she liked to learn in her own way, following her curiosity where it led, rather than following a prescribed path. As Barbara moved through her childhood years in Brooklyn, something was quietly forming inside her, a habit of mind, a way of approaching the world. She was learning to pay close attention, to look carefully at small things, to sit with a question rather than rushing toward an answer. These were not lessons anyone taught her directly. They were simply the way she was, and they would become, over time, the very foundation of her scientific life. By the time Barbara reached high school, she knew she liked science. Not in a vague way. She genuinely enjoyed the process of inquiry. The idea that you could ask a question about the natural world and then find a way to actually answer it through careful observation and experiment appealed to something deep in her. Her mother, Sarah, had a complicated relationship with Barbara's growing intellectual ambitions. Sarah was a woman of her time, shaped by the belief that women were meant to marry, to build homes, to raise families. She worried that too much education would make Barbara unmarriageable, that it would put her outside the life that was expected of women. These were not cruel concerns. They were the concerns of a mother shaped by a particular world, a world where the paths available to women were narrow and clearly defined. But Barbara was not easily redirected. She finished high school in 1919 and wanted, with a clarity that surprised even herself, to go to college. Specifically, she wanted to study at Cornell University. She wanted to study science. Her mother resisted. There were arguments. There was tension in the household. Sarah genuinely feared that a daughter with too much education would find herself alone, outside the ordinary rhythms of life that a woman was supposed to follow. Her father, Thomas, supported Barbara. Quietly but firmly, he helped make it possible for her to go. In the autumn of 1919, Barbara McClintock arrived at Cornell University in Ithaca, New York. Cornell sat on a hill above a deep gorge overlooking a wide valley. It was a place of paths through trees, of old stone buildings, of long winters and cool summers. For Barbara, it felt immediately right. It felt like a place where serious thought was possible. She enrolled in the College of Agriculture, which was one of the few divisions of Cornell where women could study science at a reasonable cost. This was a practical consideration, but it turned out to be a fortunate one, because the College of Agriculture was home to a small but serious group of people studying genetics. Genetics was a young science in 1919. The field had only recently been born. In 1900, scientists had rediscovered the work of Gregor Mendel, an Austrian monk who had spent years of the previous century carefully crossbreeding pea plants and recording what happened. Mendel had worked out the basic rules of inheritance, how traits pass from parent to offspring. His work had been ignored during his lifetime, but when it was rediscovered at the turn of the century, it ignited a new field of science almost overnight. By the time Barbara arrived at Cornell, scientists were working intensely to understand the physical basis of heredity. They knew that traits were passed down through something called genes, but they didn't know exactly what genes were made of, where they were located, or how they worked. These were open questions. Big, fascinating hours, wide-open questions. And Barbara, sitting in her first courses on genetics at Cornell, felt something click into place. This was what she wanted to do. She took her first course in genetics in 1921, during her sophomore year. It was offered through the plant breeding department, and it was one of the few places on campus where students could seriously engage with the science of heredity. By the time that course ended, Barbara had already contacted the professor and asked to be included in the graduate genetics course the following year. She was an undergraduate. That was unusual, but she asked clearly and directly, and the professor said yes. From that point forward, genetics was her world. She threw herself into the work with a focus that her classmates noticed immediately, not in a competitive way, not in a way that drew attention to itself, but in the way that someone focuses when they have found for the first time the exact thing they were meant to do. The laboratory became a comfortable place for her. The rhythms of scientific work, the careful preparation, the patient observation, the slow accumulation of evidence suited her temperament perfectly. She didn't need constant results or quick rewards. She could work steadily, over long periods, without losing her sense of purpose. Cornell in the early 1920s was a stimulating place for anyone interested in genetics. There was a small, close-knit community of researchers and students who were all working on related questions. They ate together, talked together, argued about science in hallways and over meals. It was the kind of intellectual environment that shapes people in lasting ways. Barbara found her people there. She formed friendships that would last her entire life. Among the most important was a woman named Harriet Crichton, who would later become her collaborator on one of the most significant experiments of Barbara's early career. But we'll come to that in time. There was also Marcus Rhodes, a fellow graduate student who shared her deep interest in maize genetics, and George Beadle, who would go on to win a Nobel Prize of his own. These were serious, gifted people, and Barbara moved among them as an equal, even when the world outside the laboratory didn't always see it that way. Because outside the laboratory, being a woman in science in the 1920s came with quiet but constant friction. It wasn't always overt. It wasn't always someone telling her directly that she didn't belong. It was more subtle than that. It was the way opportunities were structured, the way certain positions were simply not available to women, the way a woman with the same abilities as a man would find different doors in front of her, some open, many closed. Barbara was aware of this. She couldn't have been anything else. But she seemed, from early on, to have made a quiet internal decision not to let it consume her. She focused on the work. She let the work speak, and she moved forward. She completed her undergraduate degree in 1923, staying at Cornell to continue into graduate study. This was a natural progression for her. She was already embedded in the research community there, already doing serious work. Her graduate research focused on maize, corn, in the more common way of saying it. This might seem like an unusual choice for someone interested in the deep questions of genetics. But maize was, for geneticists of that era, an almost ideal organism to study. The reasons were practical. Maize grew relatively quickly. It produced large numbers of offspring. Its chromosomes were large enough to be visible under a light microscope, which was important at a time when scientists were trying to understand the physical structure of heredity. And the genetics of maize were complex enough to be genuinely interesting. For Barbara, working with maize meant spending long stretches of time in fields and greenhouses, observing plants at every stage of their growth. It meant learning to see, to notice subtle differences between one plant and another, to track what changed across generations. It was slow, careful, physical work as much as it was intellectual work, and she loved it. During her graduate years, Barbara began developing a set of skills that were genuinely unusual. She became extraordinarily good at cytology, the study of cells, and specifically of chromosomes, under the microscope. She could prepare chromosome samples with a level of care and precision that allowed her to see details that other researchers simply missed. This was not just technical skill, though it was that too. It was also a quality of attention, the ability to look at something very carefully for a very long time, without getting bored or distracted, to notice what was there rather than what you expected to be there. That distinction matters enormously in science. It's easy to look at something and see what you already believe. It takes a different kind of mind to look at something and see what is actually present, even when it surprises you. Barbara had that kind of mind. By the mid-1920s, she had begun making real contributions to the understanding of maize chromosomes. She developed techniques for visualizing and identifying individual chromosomes, for telling them apart, numbering them, tracking them. This was foundational work. Before you could study what chromosomes did, you needed to be able to see them clearly. And Barbara was among the first to make that possible for maize. She received her doctorate from Cornell in 1927. She was 25 years old. It was a genuine achievement, a hard-won credential in a demanding field. But a PhD for a woman in 1927 did not open the same doors it would have opened for a man. Academic positions, permanent faculty positions with stability and resources, were largely unavailable to women in the sciences at that time. This was simply the reality. It wasn't something Barbara could change by working harder or publishing more. The structure itself was the obstacle, so she remained at Cornell for several years after her doctorate, working as a research assistant and instructor. The work continued. The science didn't stop, but the path forward was less clear than it would have been for her male colleagues. During these postdoctoral years at Cornell, Barbara made what many consider to be one of her most important early contributions. Working with Harriet Crichton, the younger colleague she had befriended during her graduate years, Barbara designed a series of experiments to answer a question that had been hovering over genetics for several years. The question was this: when chromosomes exchange segments during the process that leads to the formation of egg and sperm cells, does that exchange have a physical basis, or is it only a mathematical one? Scientists had theorized about something called crossing over, the idea that chromosomes could swap segments with each other during cell division, and that this explained why certain traits appeared linked or separated in predictable ways. But no one had actually demonstrated this physically. No one had shown that you could see a chromosome break, exchange a piece with its partner, and reconnect, and that this physical exchange corresponded to a specific change in the traits of the offspring. Barbara and Harriet designed an experiment that did exactly that. They used maize plants that had specific, visible markers on particular chromosomes, features they could see, physical landmarks. This allowed them to track a specific region of a specific chromosome and watch what happened to it across generations. What they found was clear. The physical exchange of chromosomal segments corresponded directly to the inheritance of traits. Crossing over was real. It had a physical basis. You could see it. This was an important result. It provided solid, direct evidence for something that had been theoretical. It tied together two streams of knowledge: the visible world of chromosomes and the mathematical world of genetic inheritance, in a way that hadn't been done before. Barbara and Harriet wrote up their findings and submitted them for publication in 1931. The paper appeared in the Proceedings of the National Academy of Sciences. It was well received. It was recognized as important work. But even so, the recognition didn't translate easily into the kind of stable academic position that the work deserved. For Barbara, the early 1930s were a period of moving, of temporary positions and fellowships that allowed her to keep working, but never quite settled her anywhere permanently. She held a fellowship funded by the National Research Council, which gave her some freedom to travel and collaborate. She spent time at the University of Missouri, at the California Institute of Technology, at other institutions. Each place offered something: new colleagues, new equipment, new perspectives. Caltech, in particular, was an intellectually stimulating environment. There were serious researchers there, working on related questions, and the conversations she had during her time in California shaped her thinking in lasting ways. But she was always a visitor, never quite at home. This constant movement was tiring. It was also clarifying, in a way. Barbara came to understand that the academic world, as it was structured, was not going to simply make a place for her in the usual way. She would have to build her work around whatever circumstances she could find. She adapted. She continued. During this period, her work on maize chromosomes was deepening. She was developing an increasingly detailed understanding of how chromosomes were organized, how they behaved during cell division, and what happened when things went wrong. She became interested in what happened at the ends of chromosomes, regions now known as telomeres. She made observations and drew conclusions about these regions that were, at the time, largely overlooked. Decades later, when the biology of telomeres became a major area of research, her early observations would be recognized as prescient, but that recognition was still far in the future. She also became interested in the nucleolus, a small structure found inside the nucleus of cells, and its relationship to specific regions of chromosomes. Again, her observations were careful and detailed, and they pointed toward things that later science would confirm. These were not flashy discoveries. They didn't come with press releases or grand announcements. They were the slow, steady accumulation of observation and understanding. The kind of work that doesn't make headlines, but builds the foundations that later discoveries stand on. By the early 1930s, Barbara McClintock was recognized by the small community of geneticists as someone exceptional. The scientists who knew her work understood what she was doing. They knew her abilities were rare. She was elected to the National Academy of Sciences in 1944, only the third woman ever to receive that recognition. She was elected President of the Genetic Society of America. These were significant honors, and they reflected the genuine respect of her scientific peers. But esteem within a small community and a stable professional life were not the same thing, and stability was still elusive. She had spent years moving between temporary positions, fellowships that ended, institutions that welcomed her work but couldn't offer a permanent home. It was wearing, not in a way that broke her spirit. Barbara was not easily broken, but in a way that she carried quietly, day after day. Then, in 1941, something changed. Through a connection with a colleague, Barbara was invited to spend a summer at Cold Spring Harbor Laboratory on Long Island in New York. Cold Spring Harbor was, at that time, a small but serious research institution. It sat on a harbor inlet, surrounded by trees and water. The pace there was quiet. The setting was beautiful in a plain, unpretentious way. It was a place where scientists came to work without distraction. Barbara arrived for the summer. She found the setting agreeable. She found the work possible. And then she simply stayed. She was offered a research position, and she accepted. It wasn't a tenured faculty appointment at a great university. It wasn't the kind of position her abilities might have earned her in a fairer world. But it was real. It was stable. It gave her a laboratory, a field to grow her corn, and the freedom to pursue her own questions. She was 39 years old. She would remain at Cold Spring Harbor for the rest of her life. The laboratory she settled into at Cold Spring Harbor was modest. Small rooms, simple equipment, the basic tools of her trade. There was nothing grand about it, but it was hers. She could organize it the way she wanted. She could set her own hours, follow her own lines of inquiry, work at the pace that suited her. And the pace that suited Barbara McClintock was steady, deep, and unhurried. Outside the laboratory, she had her field, a plot of land where she grew her maize plants season after season. This was the heart of her work in many ways. The field was where the experiments lived, where she planted carefully chosen seeds in carefully chosen arrangements, then watched and waited across the growing season, recording what she saw. Maize takes time. You plant in the spring, and you wait through the summer, and the answers come in the autumn, when the ears are fully formed, and the kernels reveal their colors and patterns. Each growing season was one long, slow experiment, one careful question put to nature, with the answer arriving months later. Barbara had that kind of patience. Not the forced patience of someone waiting for something they'd rather have sooner. the natural patience of someone who understood that good science cannot be rushed. She lived simply at Cold Spring Harbor. She had a small apartment on the grounds of the laboratory. She didn't own much. She didn't need much. Her life was organized around her work, and her work was organized around the rhythms of her plants. She woke early. She worked long hours. She spent time in the field, time at the microscope, time at her desk thinking through what she had seen. In the evenings, she might walk along the harbor, or read, or talk quietly with colleagues. It was a contained life, a focused life, and she seemed to find in it a genuine satisfaction that had little to do with the kind of recognition the outside world offers. This is worth pausing on for a moment. Barbara McClintock, through all the years of professional uncertainty, through all the doors that had been closed to her, through all the positions she hadn't been offered, and the recognition that had been slow to come, she had not become bitter. She had not turned inward in a wounded way. She remained, by all accounts, a person of genuine warmth and dry humor, a person who laughed easily, who cared about the people around her, who was generous with her time and her knowledge. That equanimity was not indifference. She was not someone who didn't care about things. She cared deeply about the science, about the truth of what she was finding. But she had developed, or perhaps had always had, a kind of rootedness in herself that didn't depend on external approval. She knew what she saw. She knew what the evidence showed. And she was willing to wait for the world to catch up. That willingness would be tested more than she could have anticipated, because in the late 1940s and into the early 1950s, Barbara began finding something in her maize plants that didn't fit the existing understanding of how genetics worked. At the time, the dominant view of the gene was relatively straightforward. Genes sat in fixed locations on chromosomes. They were stable. They stayed where they were. They passed from parent to offspring in an orderly, predictable way. This was the foundation on which the field had been built, and it had proven enormously productive. But Barbara's corn was showing her something different. She was studying the patterns of coloration in maize kernels. The kernels of a maize ear come in many colors: yellows, purples, reds, blues, in patterns that can be spotted or striped or solid. These colors are controlled by genes, and by studying which colors appeared in which kernels and in what proportions, Barbara could learn something about what the underlying genes were doing. What she began to see was a pattern of instability, traits appearing and disappearing across generations in ways that didn't follow the established rules. Kernels showing color patterns that shouldn't have been there, given the genetics of the parent plants. She looked more carefully. She prepared chromosome samples and studied them under the microscope with her characteristic precision. And she began to see something remarkable. Pieces of chromosomes were moving, not in the chaotic, random way that damage or error might produce. In an organized, regulated way. Specific segments of chromosomes were detaching from one location and reinserting themselves in another. And when they moved, they affected the activity of the genes near their new location. These moving elements, she called them transposable elements, or transposons, could switch genes on or off as depending on where they landed. They were, in a sense, mobile regulators, not just passengers on chromosomes, but active participants in the control of gene expression. This was a fundamentally new idea. The genome was not a fixed, static thing. It was dynamic. It could reorganize itself. It had within it a system of moving parts that could respond to circumstances and alter how genes behaved. Barbara developed this idea carefully over several years. She gathered evidence season after season, building the case from the ground up, from kernel colors and chromosome observations, from careful crosses and meticulous record keeping. By the early 1950s, she was ready to present her findings to the wider scientific community. She gave a lecture at the Cold Spring Harbor Symposium in 1951. This was a significant venue. The scientists who attended these symposia were the serious researchers of the field, the people whose opinions mattered. Barbara laid out her evidence clearly and in detail, explaining what she had found and what she believed it meant. The response was largely silence. Not hostile silence exactly, not angry dismissal, but a kind of baffled quiet, a sense among many in the audience that they couldn't quite follow what she was saying, that it didn't fit the framework they had for understanding how genetics worked. Some found her presentation too complex, too detailed, too filled with the specific particulars of her maze system. They didn't have the background to evaluate the evidence she was presenting, because her work was so deeply embedded in the specifics of maze cytology that it required a level of familiarity most of her audience didn't have. Others simply couldn't accept the idea. Genes didn't move. That was a foundational assumption of the field. And here was Barbara McClintock saying not only that they could move, but that their movement was regulated and meaningful. It was too much of a departure, too far outside the existing map. She tried again. She wrote up her findings and published them. She gave further lectures and presentations. She explained and re-explained as clearly as she could, but the reception remained cool, politely skeptical at best, quietly dismissive at worst. For most scientists, this kind of response would have been devastating. To spend years building a careful, evidence-based argument, to present it to your peers, and to be met with incomprehension or disbelief, that is a deeply discouraging experience. Barbara was discouraged. She was human, and she wasn't indifferent to the response her work received. She had hoped it would land differently. She'd hoped the evidence would speak clearly enough to be understood. But she didn't collapse under the weight of the dismissal. And she didn't abandon the work. Instead, she made a quiet decision. She stopped presenting the transposon work to the broader community, not because she had lost faith in it, but because she understood that the community wasn't ready to receive it. Trying to convince people who lacked the conceptual tools to evaluate her evidence was not a productive use of her energy. So she continued the work on her own. She kept growing her maze. She kept making observations. She kept building the evidence, quietly and steadily, with no particular expectation of when or whether the world would catch up. She found other lines of inquiry to pursue alongside the transposon work. She studied the genetics of maize in Latin American populations, indigenous varieties that had been cultivated for thousands of years by the peoples of Mexico, Guatemala, and the Andes. She traveled to those regions on research trips, collecting samples, talking with farmers, documenting the extraordinary genetic diversity of maize that human cultivation had produced over millennia. This work was different in character from the cytological work she had done at the microscope. It was broader more anthropological in some ways. It connected her to the long human history of agriculture, to the ways that people had shaped plants, and plants had shaped people across thousands of years. She found it deeply interesting, and it kept her connected to the wider world of genetics, even as her transposon work sat quietly on the shelf, waiting. The 1950s passed. The 1960s came. During this time, the field of molecular biology was transformed by a series of discoveries that changed everything. The structure of DNA was determined in 1953 by Watson and Crick, building on the X-ray crystallography work of Rosalind Franklin and others. The genetic code was worked out over the following decade. The mechanisms by which DNA is copied and read were elucidated. The molecular basis of heredity, which had been abstract and theoretical for so long, was now understood in concrete, chemical terms. This was a revolutionary period in biology, and it created a new generation of scientists who thought about genetics in a completely different way than Barbara's generation had. These new scientists worked with bacteria and viruses, with molecules and chemical reactions. Their tools were different, their frameworks were different, and their relationship to the old cytological work, the chromosome-level genetics that Barbara had spent her life doing, was somewhat distant. For a time, it seemed as though Barbara's world and this new molecular world were separate streams, running alongside each other without quite meeting. But then something happened. In the early 1960s, two French scientists working at the Pasteur Institute in Paris, François Jacob and Jacques Monod, published a landmark paper. They had been studying how bacteria regulate the activity of their genes, how bacteria can turn certain genes on when they need them and turn them off when they don't. What Jacob and Monod described was a system of genetic regulation, genes that controlled the activity of other genes, a layer of organization above the simple presence or absence of a gene, a system for managing when and how genes are expressed. This idea was immediately recognized as important. It was the beginning of a new understanding of how genomes work, not just what genes are present, but how their activity is controlled. And for those who had followed Barbara's work, the parallel was striking. What Barbara had described in her transposon work, elements that moved within the genome and affected the activity of nearby genes, was a form of genetic regulation. She had been describing something very similar to what Jacob and Monod were now describing in bacteria, but she had seen it years earlier in a more complex organism, using the tools of cytology rather than molecular biology. The concepts were beginning to converge. Slowly, quietly, the scientific community began to look again at what Barbara had been saying since the early 1950s. It was not an overnight shift. These things rarely are. But over the course of the 1960s and into the 1970s, the concept of transposable elements, of moving genetic elements that regulate gene activity, began to gain credibility. Molecular biologists found transposable elements in bacteria, then in fruit flies, then in other organisms. Each new finding confirmed that what Barbara had described in maize was not a peculiarity of corn. It was a fundamental feature of genomes, something present across the living world. The evidence accumulated, the understanding deepened, and gradually, the work that had been met with silence in 1951 began to be recognized as something genuinely foundational. Barbara watched all of this from Cold Spring Harbor. She continued her own work. She continued growing her maze. She was in her 60s now, then her 70s, still in the field each growing season, still at the microscope, still thinking carefully about what she saw. She had outlasted the skepticism. She had simply continued working, year after year, and the world had eventually come to where she already was. The recognition, when it finally came in full, came with remarkable force. Awards began arriving. Honors from scientific societies. Prizes that acknowledged the importance of what she had spent her career discovering. Each one was received graciously, with the quiet, slightly amused manner that characterized Barbara's relationship to public recognition throughout her life. She had never needed the recognition to keep going, and now that it was coming, she received it without making too much of it. Then, in 1983, something happened that even Barbara could not receive entirely quietly. She was awarded the Nobel Prize in Physiology or Medicine. She was 81 years old. She was the first woman to win that prize unshared, to receive it alone, for her own work, without splitting it with a male colleague. The announcement was made in October of 1983. Barbara was in her laboratory at Cold Spring Harbor when she heard the news. Her response, by most accounts, was characteristically understated. She expressed gratitude. She said kind things about the colleagues and institutions that had supported her work over the years. And then, it is said, she went back to work. The Nobel Prize brought with it a level of public attention that Barbara had never experienced before. Journalists wanted interviews. Television programs wanted appearances. Universities wanted to give her honorary degrees. Scientific societies wanted her at their events. She handled all of it with grace. But she was also, by this point, a woman in her 80s who had spent 60 years building a particular kind of life, a quiet life, a focused life. And she had no intention of abandoning it simply because the world had suddenly decided to pay attention. She gave interviews when she felt it was worthwhile. She accepted some of the honors and declined others. She continued to show up at Cold Spring Harbor, to walk the grounds she had walked for decades, to spend time in the laboratory that had been her home for so long. She was not reclusive. She had never been reclusive. She enjoyed conversation and connection with people she found interesting, but she was deeply private in the sense that her inner life was her own, and she had little interest in performing herself for public consumption. What she did do, in the years after the Nobel, was talk more openly about her philosophy of science, about what she believed good science required, about the habits of mind and spirit that she thought were necessary for real discovery. She spoke about the importance of what she called a feeling for the organism. This phrase, simple as it sounds, carried a great deal of meaning for Barbara. What she meant was this: to do good science, it is not enough to run experiments and record results. You have to develop a genuine relationship with what you are studying. You have to spend enough time with it, look at it carefully enough and long enough that you begin to understand it from the inside. You begin to sense what it might do, not because you've calculated it, but because you know it. This sounds almost mystical, and Barbara knew that it sounded that way to some people. But she was not describing mysticism. She was describing a very deep and very patient form of attention. the kind of attention that allows you to notice things that a more superficial observer would miss entirely. She had developed that feeling as her corn plants over decades. She knew them in the way that a person can know another living thing after years of careful, attentive company. And that knowledge had guided her toward questions and observations that no one else had been positioned to make. She believed this kind of knowledge was undervalued in science, that the drive towards speed and efficiency and large-scale data gathering was, in some ways, pulling scientists away from the slow, patient, intimate attention that leads to genuine discovery. It was a thoughtful critique, and coming from someone with her record, it carried weight. She also spoke about the experience of being dismissed, about the years when her transposon work was met with incomprehension. She spoke about it without bitterness, which struck people who heard her as remarkable. She seemed to have genuinely processed that experience and come out the other side with her sense of self intact. She said that she had always known what she had found. She had been certain of the evidence, and that certainty had been enough to sustain her through the years when no one else seemed to see it. This is easier said than lived. Most people, facing sustained dismissal from their professional community, would either abandon the work or be consumed by resentment. Barbara had done neither. She had simply continued, with patience and with a kind of internal steadiness that seemed to come from somewhere very deep. Some people who studied her life later tried to understand where that steadiness came from. What had given her such an unusual capacity to persist without external validation? Part of it, certainly, was temperament. Barbara had always been self-contained. Even as a child, she'd been comfortable in her own company, comfortable following her own direction. She hadn't needed the world's approval to feel confident in what she was doing. Part of it was also, perhaps, the nature of the work itself. She was not theorizing in the abstract. She was looking at actual organisms, actual chromosomes, actual patterns in actual kernels of corn. The evidence was in front of her, season after season. It didn't go away because other scientists didn't believe in it. The corn kept showing her what it showed her. And part of it, perhaps, was a kind of philosophical acceptance that she had developed over the years. An understanding that truth and recognition do not always arrive together. That the value of a discovery is not diminished by the fact that it takes the world a long time to understand it. She had made her peace with that, deeply and genuinely, it seemed. In the years after the Nobel Prize, Barbara continued to be a presence at Cold Spring Harbor. She attended seminars. She talked with younger scientists. She asked questions that were still sharp, still penetrating, still coming from a mind that had lost none of its essential quality. She had grown old in the way that some people grow old who have lived close to their own nature for a long time. She was smaller physically, more fragile in the ways that the body eventually becomes fragile. But her eyes were still clear and direct. Her manner was still warm and occasionally quite funny. She still had opinions, and she still expressed them without much concern for whether they were the expected ones. Young scientists who had the chance to spend time with her in those final years often described the experience as quietly extraordinary. She didn't lecture or perform. She simply talked. She asked about their work with genuine curiosity. She offered observations that were sometimes surprising in their originality. She was still a person whose mind moved in its own particular direction. She had never married. She had never had children. This was a choice, or perhaps more accurately, a natural expression of the way her life had organized itself. She'd poured herself into her work and into her intellectual relationships, and those had been enough, more than enough. She had close friendships that lasted decades. She had colleagues who became genuinely dear to her. She had the long company of her plants, her laboratory, her field. She had Cold Spring Harbor, with its water and its trees, and its particular quality of quiet. It was a full life, unusual in its shape, but full. As she moved through her late 80s, Barbara's health gradually declined in the ordinary ways. She moved more slowly. She tired more easily. She spent less time in the field, less time at the microscope, but she remained present. She remained herself. She died on September 2nd, 1992. She was 90 years old. She died at Cold Spring Harbor, in the place she had made her home for more than 50 years. Quietly, after a short illness, in the autumn, when the corn in the fields would have been nearing the end of its season. It was a peaceful ending, appropriate in its way to the life she had lived. The scientific community responded to her death with genuine mourning. Tributes came from researchers around the world, from the great institutions of biology and genetics, from colleagues who had known her for decades, and from younger scientists who had only encountered her work through papers and lectures. What they all described in different ways was the same thing: a mind that had seen further and deeper than almost anyone else of her generation. A scientist who had been willing to trust what the evidence showed her, even when the world wasn't ready to follow. Her discovery of transposable elements, those moving pieces of the genome that she had first observed in her maize fields in the 1940s, had by the time of her death been confirmed in virtually every living organism that had been studied at the molecular level. From bacteria to human beings, genomes were filled with transposable elements. These elements made up, in fact, a substantial portion of the human genome. They were not quirks or errors. They were fundamental features of how genomes are organized and how they function. The implications of this were still being worked out at the time of her death, and they are still being worked out today. The biology of transposable elements has become a rich and active field of research, touching on questions about evolution, about gene regulation, about the causes of disease, about the origins of genetic diversity. All of it traces back, in some sense, to a woman in a field in New York, looking carefully at the colors of corn kernels, season after season, in the 1940s and 1950s. It is a remarkable lineage. Beyond the specific discovery, Barbara McClintock left something else, something harder to measure, but perhaps equally important. She left an example of what it looks like to pursue a question with complete honesty and complete commitment, to follow the evidence wherever it leads, even when it leads somewhere that the established understanding cannot yet accommodate, to do the work for the sake of the work, without requiring that the world immediately recognize its value. This is not a common way to live. It requires a kind of courage that doesn't announce itself loudly, not the courage of dramatic moments, but the quieter courage of continuing, of showing up year after year, to the same field, the same microscope, the same careful questions. Barbara had that courage. She had it in abundance, and she exercised it without drama, without complaint, and without asking anyone to notice. The Nobel Committee, in 1983, had called her discovery one of the two major surprises in genetics of the 20th century. The other, they said, was the discovery of the double helix structure of DNA. To be placed in that company, with work done decades earlier, largely alone, without institutional support or professional recognition, it says something about the magnitude of what she had accomplished. And yet Barbara herself never seemed to think in those terms. She didn't compare herself to others. She didn't rank her achievements. She had found something true, and she had followed it faithfully for 50 years. And that was what mattered to her. It is worth spending a moment with that. In a world that is very good at measuring and ranking and comparing, there is something quietly radical about a person who simply does the work because the work is worth doing, who finds meaning in the daily practice of careful attention, rather than in the recognition that attention might eventually produce. Barbara McClintock spent her life doing something very simple, in its way. She looked at her corn plants. She looked very carefully, for a very long time, and she saw something that no one else had seen. And then she waited, with patience that most of us can barely imagine, for the world to learn to see it too. That is her story, or the shape of it anyway. The full texture of a life lived that carefully, that quietly, over 90 years. That is something that can only be approached from the outside, suggested rather than captured. But perhaps that is enough. Perhaps it is enough to know that such a life was lived, that someone moved through the world with that quality of attention, that depth of commitment, that steadiness of spirit. Perhaps it is enough to rest in the knowledge of it and to let it settle quietly, like the end of a long growing season, when the fields are still and the harvest is in and everything is calm. You've been listening to Bedtime Biographies. Thank you for spending this time with the life of Barbara McClintock. We hope the story found you well, and that wherever you are, you're comfortable and warm and ready to sleep. The day is done. The work is done. Let everything be still now. Good night.